Okay, good afternoon, good morning, good evening to everyone, and greetings to all of you for joining us from any part of the world that you might be. My name is Lamarana Jallo. I am a PhD student in biochemistry at the University of Ghana, West African Center for Cell Biology of Infectious Pathogens, and the American Society for Microbiology Young Ambassador to the Gambia. It is truly my pleasure to welcome you to this ASM webinar, which is jointly organized by ASM Young Ambassadors in partnership with supporting institutions. I'd like to take a moment to acknowledge the organizing partners, which are myself, representing the Gambia, and the ASM Young Ambassador to China, who also happens to be our guest speaker for today. The Antimicrobial Resistance Group at the West African Center for Cell Biology of Infectious Pathogens, which is led by Dr. Abiola, a faculty member at the department, and the Gambia Society for Microbiology. And since this is the first of our Young Ambassador webinar series, I would like to give a bit background as to what the webinar is about. The purpose of this platform is to foster knowledge exchange and collaboration, both within the West African region and between Africa and Asia, particularly Africa and China, Gambia, Ghana, and China, and indeed the broader global scientific community, all in the spirit of advancing microbial sciences to drive positive impact for humanity. In today's interconnected world, collaboration is not just a benefit, it is a necessity. Our hope is that this initiative will serve as a catalyst for meaningful contributions and innovation in microbiology and science as a whole. Now, it is my honor to introduce our guest speaker, Dr. Ze Li, as I've said initially. He is the ASM Young Ambassador to China. Dr. Li is a principal investigator and an attending physician at the third affiliated hospital of Sun Yat-sen University. He completed both his master's and PhD degrees in obstetrics and gynecology at the University of Jinan, China. Dr. Li brings with him a wealth of academic and clinical experience. His professional background includes, excluding the postdoctoral work, his professional background includes serving as an attending physician at the Department of Obstetrics at the third affiliated hospital of Sun Yat-sen University. Dr. Li has contributed significantly to high-impact scientific literature in maternal and neonatal health. And to save time, I won't go through all his publications, but they speak volumes of his dedication to scientific advancement. And today, Dr. Li will be sharing with us an exciting and highly relevant presentation on the topic, the role of gut microbiota and prebiotics in maternal and neonatal health. And the webinar, basically the format is, it's going to run for 1 hour. There will be 40 minutes for the presentation, and then we'll have 20 minutes for questions and answers. And we highly encourage everyone to ask as many questions as you would have. I'm sure Dr. Li would be happy to answer all of them or any comments that you might have. Now, with that being said, it is my great pleasure to now hand the floor over to Dr. Li. Please, Dr. Li, you are most welcome. The stage is yours.
Okay, thank you for your introduction, Dr. Lamarana. Hello, everyone. I'm Dr. Li Zhe. It's said in Chinese. My name is Dr. Zhe Li from China. I'm the young ambassador to China for American Society of Microbiology. Good afternoon and good evening, everyone. I'm an obstetrician and researcher from the third affiliated hospital of Sun Yat-sen University in Guangzhou, China. It's my great pleasure to be here with microbiologist clinicians and students around the world, especially from Gambia, to share some of our research on a topic that bridges obstetrics, microbiome science, and clinical nutrition, the role of gut microbiota and probiotics in maternal and neonatal health. Over the next 25 to 30 minutes, I'll present insights from our randomized controlled trials we've conducted, focusing on how probiotic interventions during pregnancy may reshape the microbiota in mothers and their infants, and what this might mean for clinical care and long-term health. So let's restart. As we know, the human body is the host to trillions of microbes, and during pregnancy, this microbiota goes through significant changes. What's more, you don't just stay with the mother. The maternal microbiota, particularly from the gut, vagina, breast milk, and even the placenta, gets transmitted to the infant and helps to establish the early life microbiome ecosystem. This early colonization is essential. It plays a vital role in programming the newborn's immune system, metabolism, and even neurological development. When this colonization process is disrupted, whether by cesarean section delivery, antibiotics, or other factors, it has been associated with an increased risk of allergic diseases, obesity, inflammatory conditions, and neurodevelopmental challenges. So, understanding and supporting the maternal-infant microbiome connection is critical, very critical. Why we consider probiotics interventions during pregnancy? Because probiotics are defined as a live microorganism that, when administered in adequate amounts, confer a health benefit on the host. They are already widely used to support, for example, gut health in the general population, and studies show they're safe and well tolerated during pregnancy. But what we are now learning is that probiotics may have much broader impacts. They can enhance the functional diversity of the maternal microbiota, help regulate immune responses, and may even reduce the risk of gestational diabetes, preeclampsia, and infant allergic diseases. Importantly, probiotics may help train the maternal microbiota in a way that improves vertical transmission, benefiting the infant's gut colonization and long-term health. This is the foundation of our research, using probiotics as a tool to optimize maternal and neonatal outcomes by reshaping the microbiome. Before we dive into the individual results, let me give you a quick overview of our study framework. Between 2019 and 2024, we conducted randomized controlled trials involving healthy pregnant women and their babies. In our study, participants in the intervention group received probiotic tablets from 32 weeks of gestation until delivery. This contained a combination of Bifidobacterium logum, Lactobacillus bulgaricus, and Streptococcus Pseudomonas. We collected a wide range of biological samples, for example, maternal feces, vaginal secretions, placenta, breast milk, and infant stool at various time points, from late pregnancy all the way through 6 months postpartum. This comprehensive sampling design allows us to track the maternal microbiome changes, vertical transmission patterns, and the development of neonate microbiota with and without probiotics intervention. The following slides will walk you through our study's unique focus and findings. Let's begin with our first study. In our first study, published in Microbiology Spectrum, we looked at how probiotics influence the functional potential of the maternal gut microbiota. We enrolled 32 healthy pregnant women, randomly assigned to receive either probiotics or no supplementation from 32 weeks of gestation until delivery. Instead of looking at the microbiome composition, we use the pig rats to predict the functional genes present in the gut microbiota. The probiotic group showed a significant increase in genes associated with energy production, folate synthesis, and biotin metabolism. On the other hand, we observed a decrease in genes linked to infection disease pathway and vitamin B6 metabolism. This suggests that probiotics may support beneficial metabolic functions while suppressing pathway associated with inflammation or infection. Interestingly, genera like Bloschia, Ruminococcus, and Subdoligranulus lulam were also significantly enriched. These bacteria are commonly associated with a healthy, stable gut and ecosystem. This is our first study. And our second study focused on how probiotics may regulate immune status during late pregnancy. As we know, pregnancy requires delicate immunological balance. Early pregnancy favors anti-inflammatory tolerance and to support the fetus, while late pregnancy must prepare for inflammation driving labor. In this randomized controlled trial, we measured inflammatory cytokines in the maternal blood before and after probiotic supplementation. We found significant increase in interleukin 1 beta, interleukin 2, interleukin 12, and interferon gamma in the probiotic group. This shift indicates a move from a tolerogenic state toward a mild pro-inflammatory readiness, which may help the body prepare for the inflammatory processes necessary for labor. This result suggested that probiotics don't just act on gut microbes, they also have meaningful effects on maternal immune adaptation near term. So this is our second study. We also have the third study. The third study published in Frontiers in Nutrition aimed to trace the origin of neonatal gut microbiota over the first 2 weeks of life. We collected samples from 30 mother-infant pairs, maternal gut, vaginal tract, and placenta, and from neonates on day 1, day 3, and day 14 after birth. We're using substructure and find that the placental microbiota had the strongest similar to day 1 mechanism, suggesting intrauterine microbiome influence. By day 3 and day 14, the maternal gut microbiota became the primary contributor to neonatal gut colonization. Interestingly, although the probiotic inventation did not significantly increased. Bifidobacterium in maternal samples, it did reshape the overall structure of both maternal and infant microbiota. These findings support the concept of a dynamic, stage-specific maternal-infant microbiota connection and highlight that even subtle shifts induced by probiotics may have long-term effects. The fourth study, published in Highly Young, we explored a new angle, how maternal probiotics affect not just the mother's gut, but also breast milk and whether this in turn influences the infant's gut microbiota. We recruited 27 pregnant women who received probiotics or no intervention starting at 32 weeks. Breast milk and infant fecal samples were collected when babies reached 6 months old. Our analysis revealed that probiotics led to a distinct milk microbiota with 9 bacteria uh, geners enriched and 6 reduced compared to controlled. The CM means control maternal group, and PM means probiotic maternal group, and the CI means infant in control group, and the PI means infant in probiotic group. Even more compelling, we observed a mirror shift in the infant gut microbiota. 14 geners were more abundant in infants whose mother had taken probiotics. Spearman correlation analysis showed that several bacteria geners in milk were positively associated with those in infant gut, suggesting a vertical probiotic connection. So that means even mothers after delivery, probiotics take during pregnancy may have long-lasting microbial effects on the child through the bridge of breast milk. The last but not the least, this study brings everything together. Here we examined how probiotics affect not just one site, but multiple maternal microbiota compartments, the gut, the vaginal tract, and the placenta. By comparing this with the neonatal gut microbiota across 3 time points, we could map out how different maternal sites contribute to infant colonization. In the probiotic group, we observed the changes in microbial structure, not just composition, in all maternal compartments. Certain bacteria like Valonia and Romnicoccus were altered in maternal gut, and similar patterns appeared in neonatal meconium. Interestingly, the actual probiotic strains we administered, like Bifidobacterium logum, were not significantly increased in the sample. We think this implies that probiotics may not take over the system, but instead shift the ecological balance, possibly by supporting the growth of beneficial and generous microbes. This ecological multi-site interaction is likely key to understanding how perinatal microbiota interventions truly work. At this point, I'd like to bring everything together. Across our clinical studies, we've mapped how probiotics impact that the maternal microbiota at different body sites, and how these sites influence the infant gut ecosystem. The maternal gut responds first, with improved microbial function and immune redness. These changes ripple across to the placenta, vaginal tract, and even breast milk, creating an interconnected microbial network. The neonates then received the microbial legacy through delivery and breastfeeding, gradually shaping their own gut microbiota. This systems level view helps explain why perinatal probiotics, even for a short period, may lead to long-term health programming. It's not about one bacterium. It's about orchestrating a healthier microbial symphony across the mother-infant axis. So, what do all these microbial shifts mean in a real-world clinical setting? First, we can confidently say that probiotic use during late pregnancy is safe and well tolerated. For mother, probiotics may help reduce the risk of gestational diabetes, constipations, and even aids in postpartum recovery. By the way, this has been showed in previous work by other studies. They encourage, they enhance gut microbiome function and promote a more favorable immune environment near delivery. For infants, maternal probiotics can indirectly improve early gut colonization, especially through milk and placental pathways. This may lower the risk of allergies and support immune maturation. From a translational perspective, these studies support the development of targeted microbiome interventions. Perhaps even personalized probiotics tailored to a women's microbiome profile. In short, modulating the microbiota isn't just a scientific idea anymore. It's becoming a clinical reality in perinatal care. As with any clinical research, there are some important limitations to acknowledge. First, our studies had a relatively small sample sizes, most with 27 to 32 participants, due in part to strict inclusion criteria like exclusive breastfeeding and no antibiotics. Also, the probiotic intervention began in the third trimester. It remains unclear whether earlier or longer term supplementation could have stronger effects. We used 16 as RNA sequencing, which limited to our ability to detect strain-specific colonization or full functional uh, potential. And also, the probiotic species wasn't significantly enriched. Uh, we observed the ecosystem level changes, suggesting that microbiome modulation may work through indirect ecological effects. Looking forward, we hope to scale up this study across different populations, apply more comprehensive multi-omics, and ultimately work toward personalized probiotic strategies for mothers. Most importantly, we aim to follow up on infant health outcomes, including incidence, neurodevelopment, and metabolic markers in the first year of life. At last, I'd like to sincerely thank all the incredible people and institutions who made these studies possible. First, I'm grateful to Professor Xiao for her mentorship and scientific guidance. Specific thanks to go to the clinical terms, lab technicians, and bioinformatics experts who supported us behind the screen. I also want to thank the funding agencies, including the National Natural Science Foundation of China and the Guangdong Province Medical Science Program for supporting this research. And most of all, I thank the mothers and the newborns who participate in our trials. They made this research possible. Thank you so much for your attention and your time. I'd like to hear your thoughts or questions. Please feel free to ask now or reach out later via email or social platforms. If you'd like to collaborate, discuss any of the topics I covered, or learn more about our ongoing work, I'm happy to connect it. I'll end with a thought. Microbes are not just the passengers, they are co-pilots in maternal and neonatal health. Thank you again. Thank you for your attention.
Um, Thank you so much, Dr. Lee, for that truly enlightening and thought-provoking presentation. You've taken us on a fascinating journey through the intricate interactions between maternal microbiota, probiotics, and neonatal health, all grounded in strong clinical evidence and translational potential. One aspect that particularly captivated me was your emphasis on strain-specific probiotic effects, especially how different strains can influence outcomes like microbiota composition, maternal inflammation, and infant development in distinct ways. It's a very powerful reminder of how precise and intentional our approaches in microbiome research must be. Now, with that foundation laid, we will now open the floor for questions and discussions. Please feel free to post your questions in the chat or Q&A box, or you can even directly ask your question to Dr. Lee. We'll try to address as many as possible within the next 20 minutes. Thank you.
Okay.
Okay, Dr. Bajinka has raised a hand. Yes, please go ahead, Dr. Bajinka. You can ask your question. Yes, Dr. Bajinka, please, you can go ahead and ask your question. I'm not sure if you are having difficulties in posting the question or what. Okay,
Okay, I'll show my email, and if you have any questions, you want to connect with me or ask me, you can through email.
Okay. You can please type your email on the comments box, Dr. Lee, so everyone can get it there.
Okay. I'll share my screen, the last of
Okay.
last slide.
Perfect. This does very well. So while people are thinking of question to ask, I just have one question. I noticed most of
Okay, please.
our attendees um basically not within this niche of gut microbiome research. Many people registered, but perhaps for some reason most were not able to join. But I have this question. You spoke a lot about the importance of gut bacteria during pregnancy and after birth. For people come who don't come from a science background, or perhaps people who are not within this area of science, could you please explain in simple terms how a mother's gut health? I believe with gut health you meant the different types of microbes that are within the mother. So could you please explain in simple terms how the mother's gut health can actually affect her baby's health even before the baby is born?
Um, Actually, the maternal microbiota, how to influence the baby's microbiota is not very clear. Um, there are so many research focusing on this because there are so many factors will affect this influence. For example, the delivery method, cesarean section and natural birth is different. And if or if not, the baby had uh, breastfeeding, their gut microbiota is different, and the region is different. The environment they live is different. So it's very hard to say how exactly the mother's gut microbiota can directly influence the fetal's uh, microbiota. But we found that actually, uh, for example, mm, if the baby uh, get birth with natural birth, they will go through with the vagina, right? Maybe before our study, we thought that the uh, vaginal microbiota will influence the baby most, but after this, we found that maybe the vagina is not the most important thing to the baby's gut microbiota. Maybe the uh, maternal gut microbiota is the most important thing for baby's microbiota. But how? Because the maternal gut and baby's gut is not connect. How? So this is the future most important thing we should do to find the connection between maternal gut and baby's gut. And really, and truly say that probiotics is very important, but in some regions and in some countries, probiotics is not widely used. But something can help to uh, let the maternal gut microbiota more uh, healthy, like yogurt, uh, like let me see how to say in English. Yeah, like yogurt and like soya cream, like something like this. Maybe I don't know what's the situation in your country, but in China, if the uh, if a woman is pregnant, they uh, during the perinatal period, they they all like use yogurt because constipation is very common in the perinatal period. The yogurt can can help them to deal with the constipation. So it is a simple method to uh, get our gut microbiota more health because yogurt is uh, full of some uh, probiotics. Yeah.
Wow. Thank you so much for that answer. It's very enlightening. I'm sure our participants would take along something with that. So okay, we have someone raising hand, but I said to Damfa. So before that, I have a question from Adam. I'll read that to you, Dr. Lee. She said, "How does an imbalance in maternal microbiota affect fetal development?
Um, how does imbalancing per maternal? Okay. Uh, because in perinatal period, the microbiota is very varied during all the whole period. From the first trimester, the second trimester, and the third trimester, the gut microbiota varied during the whole period. And in maternal, the maternal microbiota during the perinatal period will uh, will face face like antibiotic use usage, and will face like cesarean section, and will face like um, some medicine used. This all things will influence the maternal microbiota. And if the in this gut microbiota become imbalanced, the fetal will face more allergic disease, because now we found that the first year of the fetus, not the fetus, neonate, yeah, the neonate, in the first year of life, the all the uh, 2 years of life, they will challenged more allergic disease because of the imbalanced maternal microbiota. But this is the one of the challenge the fetus will face because of the imbalance of maternal microbiota. Other things, for example, just as I say in my slides, for example, neurodevelopment challenge and other things, just like this. So now we focused on how the neonate disease with the maternal microbiota, including gut microbiota, vaginal microbiota, and now the placenta microbiota, just like this. I hope my answer can satisfy with you. Yeah.
Oh, that's wonderful. Thank you so much for that enlightened answer. So I saw two Damfa's hands were up. Please, Madam Damfa, you can ask your question. Let me see if I can give you the chance to. Should Okay. You can ask your question or you can type it directly for. Okay. Thank you. Okay. While Okay, thank you so much, Danvas. You posted a comment. Thank you so much for the presentation. Thank you, Danvas.
Actually, I have a question. Actually,
Okay.
because gut microbiota in Africa is not very popular.
Yeah.
Actually, I have to say, especially in perinatal microbiota.
Yeah.
So if you want interested with this topic, uh, please you can collaborate with us, because we have technicians, we have the uh, we have uh, a team can work with this, and we can collaborate if you're interested with these topics, because future is the whole world, I think, and the neonate is our future. If we want to our neonate more health, uh, the gut microbiota of maternal and neonate is very important.
This is excellent. I must applaud this. I am sure we have many faculty members in this platform. They will be interested in collaboration, especially interregional collaboration. So this is something that would be enticing. So since they have your email, I am sure they would contact you. So you would surely hear from them. We Dr. Bajinga has just posted his question. Usman Bajinga. He's also very active in gut microbiome research. He said birth through vagina is important for the baby's microbiome, but the Chinese prefer C-section. Can you elaborate on this, please? So, um, this guy, Dr. Bajinga, did his PhD in China, so I'm sure he's a bit familiar with Chinese culture. So, can you elaborate on this, please, if you got the question?
Actually, as I know, the Chinese cesarean section rate is not very high. It's just about 30% or 40%. It's lower than the whole world cesarean section rate. And some scholars thought that the uh, birth weight, like natural birth or the cesarean section, is important to the neonate gut microbiota. This, uh, this topic is, um, how can I say? This topic is controversial. This topic is controversial because some scholars thought that the birth method is not cannot influence the neonate microbiota. So I think further study is important to deal with these controversial views. Yeah.
Okay,
Yes.
that's fine. I'm sure Dr. Bajinga have got that answer. So we had another person raise the hand, Umar An. Umar An, you can post your question, please, or you can
Yeah, I
directly
saw
ask.
this. It is surprising that the vaginal microbiota was not significant, but the gut despite. Yeah, it's possible. Um, uh, I will show my slides again about this study because our this study, yeah, our this study showed that maybe the uh, this is placenta. The placenta maybe is vital to the first day of physis, the first day of microbiota of neonate, and du during the time past, uh, the most important thing to neonate microbiota is the maternal gut microbiota. Yeah, the vaginal microbiota is not very important to babies. This is our findings, and I saw some previous work by other scholars also show this result. But some scholars showed that uh, vaginal microbiota is very important. So this is also a controversial view. So I think further studies with different regions, different countries, and different uh, people, and more larger sample size is needed to uh, deal with this controversial view.
Yes, I would also agree with that. We've done a bit of work on the gut microbiome at my department here. And what I noticed is that the microbiome is very specific, even to the individual. And across cultures, you might have different disparities. So it will be very important to have
Yeah,
perspectives based on different geographical location and different ethnic backgrounds. So
yeah.
we have this question here from Damfa. He said, is it significant in the sense that you have probiotics?
I beg your pardon. I can't get your this question.
Okay,
What
okay. The
is your question?
question,
Is it
the
a
question,
sentence you have?
yeah.
Yeah, is that you have probiotics. Uh, what does this mean? Is it significant in the sense that you have probiotics? I can't get this meaning of this sentence. What does it mean? Is
Oh,
it?
okay. She posted there was something before that. It just came to my attention. The question says, I just wanted to ask about the significance of giving tetracycline ointment as a prophylaxis for protecting against neo neonatorium conjunctivitis. I'm not sure if you are.
Makes sense. Oh, okay, I saw. Mm, maybe this question is not my it's not the scope of my study, so I can't uh tell you more details. My focus is just about the perinatal microbiota, the medicine for this neonatal conjunctivitis is not my scope of study. Sorry, I can't answer this question.
Oh, okay, okay, that's fine. I'm sure. I know Dr. Dava is a nurse, so she'll be very interested in how this thing would play out in her disciplines. So we're having questions. So while the questions are coming, I would want to move on to one tangent. You did mention about collaborations. So I would ask this on behalf of those that are speaking and who are interested in collaborations, especially with China. So China has advanced sequencing platforms and trial infrastructure, whilst Ghana and the Gambia, so we have players from Ghana also because the Workbib Center is from Ghana. Ghana and the Gambia have unique patient population and field trial experience. Now, when we bring these strengths together, it obviously is more than capacity building, it is knowledge exchange. So, in your view, what are the key ingredients needed to initiate an China-Africa research collaboration that is equitable and mutually beneficial?
Yes, I saw another question. You mentioned allergies while answering my first question. Can alternation in neonatal microbiota increase to disease like asthma and allergies? Yeah, yeah. Now there are many researchers focusing on this your annual question because neonatal microbiota increased susceptibility to disease like the allergies and the asthma, and they give the neonate probiotics to defend this, to deal with this. Yeah, and we thought that this is the neonate after birth. So we want to change the situation before birth because we can give the maternal give the mother probiotics to deal with it, and we thought this probiotics can influence the neonate after birth and decrease the susceptibility to diseases like asthma and something like this. Yeah.
Perfect. That's wonderfully answered. So would you be able to say something in what I was just alluding to question? Can you hear me, Dr. Lee?
Yeah. Yeah.
Yeah. I said, what can you say about the question I asked prior in terms of the collaborations that you just mentioned here? In your view, what are the key ingredients needed to basically initiate a China-Africa collaboration that is equitable and mutually beneficial? We understand China has an advanced sequencing, platform, trial infrastructure, and all those things. And Gambia and Ghana is a bit, well, we don't have what China have. So in your view, what are they? how do we, if somebody is interested, how probably would they navigate this?
For my view, we have some randomized clinical trial like maternal gut microbiota, how to uh, how to influence the neonatal gut microbiota and the probiotic intervention. But this is just the Chinese people. We can use this topic in Gambia, like Africa people, uh, in different country, in different regions, with different people. We can use our randomized trial to your country. So if you have the sample, we can help you to get all the results with our results. For example, China and Gambia, all the results will come out, and we can fix this, and we can tell the people, um, it's not just the Chinese people. The Chinese pregnant women were influenced by the probiotics, and these probiotics will influence the neonate gut microbiota. The Africa people, the Gambia people, is also can influenced due by this. Yeah, I think this is mm, the most important thing we can collaborate. And in the future, if you have any ideas about the microbiota in perinatal period, you we can we can talk, we can collaborate in different country, in different regions. Yeah, I think that this is my view.
Excellent, excellent. That's very good. But I just want to basically still on that same question. We know that money makes the world go around. Funding is very important. So do you see the possibility of funding coming into this in terms of getting the work done from even if it's sample collection or any other aspect within Africa or Ghana? Do you see funding coming into this area of collaboration, basically?
Yeah, I think it's possible because in our country there are some collaboration funding that can help the Chinese scientists and the other country scientists to work together to collaborate and to have one study and or some studies or several studies. We in our country there are funding like this. So if we have ideas about the perinatal microbiota, we can collaborating and to get this funding. It's possible.
That's very wonderful to hear. That's very well.
Yeah.
So since there is no more question and we are almost at the end of the presentation, I guess we will be wrapping up. So thank you so much, Dr. Lee, for that insightful and thought-provoking presentation. Your work has clearly sparked interest, and I know our attendees are walking away with valuable knowledge on the gut microbiota's role in maternal and neonatal health. To our attendees, thank you all for joining us from near and from far. Your engaging questions and presence made today's session truly enriching. This is a series, so we will be having subsequent webinars with other experts in different fields of microbiology and science as a whole. And a special thanks goes to the young ambassadors of Africa and Asia, the AMR group at the Work Group Center, the Gambia Society for Microbiology for their partnership in making this webinar possible, and to the American Society for Microbiology National for hosting this webinar on their official Zoom account. So yes, Dr. Lee, if you have any final words, please, the floor is yours.
Okay. Thank you again for your invitation, and thank you again for all the audience standing here to share with me your questions and pay your attention to my work. I hope if you have any question about my work and if you have any questions about the collaborations, you can uh, con contact me. And my email, I will type into the uh, chat column, and you can uh, chat with me uh, anytime, anywhere. Okay?
Thank you so much. That's very wonderful. To anyone who is interested in a collaboration, you can directly reach out to Dr. Lee, or alternatively, you could involve me, and then we could see how best to actually get this work. I would be happy to actually help facilitate this. Yes.
Yeah.
Um.
Thank you.
Thank you so much. So I'm going to end the recording now. Have you posted your email? Okay. Very well.
Yeah, awesome. Yeah.
Very well. So I am going to end the recording now, and thank
Uh,
you.
Now
Yes.
there are just a typo. Sorry, sorry. I'll check this.
Okay. Okay. Feel free.
Uh, the last one is the correct one. The mail dot sysu dot edu dot cn.
Yeah.
This
Perfect.
is my email.
Perfect. I'm sure they would contact you. So thank you so much again. Thanks you once again, Dr. Lee, and thank you to our presenters. It was a very wonderful session with you all. See you during our subsequent webinar.